Today we will be presenting our mystery molecule project. Our team, Carter and Matthew, have been working to explore the identity, function, and medical importance of our sequence. Over the next 10 minutes, we will guide you through our findings, including the gene structure, its function, and its impact on human health. So, for slide 1, we began with identifying our mystery sequence. We used the BLAST database, which confirmed that our sequence was retinoblastoma 1, which is from humans. The RB1 gene is found in chromosome 13, and it has about 27 sections that are called exons. It produces the retinoblastoma protein, PRB, which helps control the cell growth. And this protein acts like a brake. It stops cells from growing too much and dividing too quickly. This protein, when it's missing or has a harmful change or mutation, it allows the cell to grow out of control. This leads to increased mutations and can lead to cancer in a lot of cases.
Some of the alternative designations are bilateral retinoblastomia, diseases in both eyes, and unilateral retinoblastomia, disease in one eye. They're about the same probability-wise between them.
So signs and symptoms and prevalence would be white glow in the pupils during flash photos. You could see crossed eyes, bad eyesight, swelling, but those are a little less common. It does cause a lot of serious health problems if it has a mutation or isn't there. So a rare but fast-growing eye cancer.
And some of the symptoms, signs, and prevalence of retinoblastomia is leucario. It's the most common sign of retinoblastomia, and it's the white glow in pupils during flash photos, which you can see over here on this child. They have that, which is a clear indicator and the most common one of retinoblastomia. Starbius, crossed eyes, and some of the less common ones that you see but are still prevalent are bad eyesight, redness, and swelling of the eye. Like I said, less common, but you could still see them with people who have retinoblastomia. The prevalence and demographic of the people who generally have retinoblastomia. So the prevalence is it's a rare eye cancer. So 1 out of 15,000 births worldwide will have retinoblastomia. Will have or will develop retinoblastomia. There's 8,000 new cases every single year globally. So somewhat rare, somewhat not, if you really think about it. But the demographic, it's mainly children before the age of 5. Most are diagnosed before 24 months, and it's really a disease that you see in children, really young children. Patient outcome. So the patient outcome is very heavily dependent on when it was detected, the access to healthcare, and if you're in a developed compared to an undeveloped country. So developed countries, if detected early, detected and treated early, you have a 95% survival chance, which is pretty good for a cancer, But for non-developed countries, it's 30 to 50% survival rate, mainly because delayed diagnosis, they have limited access to healthcare, and if you don't treat it early, and if you don't try to, do that, then it just drastically goes down your survival percentage. Some of the treatment options for retinoblastomia are chemotherapy, laser therapy, which you can see right here. This would be an example of laser therapy in the terms of retinoblastomia. So laser coming down, it's destroying the cancer cells, which we showed, the cancer that we showed in the previous one. As you can see, it's destroying that. So that's kind of how the laser one works. There's also cryotherapy, freezing, and in extreme cases, removal of the eye, because it's better to just get rid of the eye than it to spread to the rest of your body and you die. So lifelong follow-up is needed after overcoming retinoblastomia. After you overcome it, you have to go into the doctor every so often because they want to make sure that the cancer doesn't come back, because that's something cancer does. The morbidity, a lot of times it comes with the cancer spreading to a more vital region of the body. So that's really how it does grow. It originates in the eye from an unregulated cell growth, and then it spreads to other parts of your body that are more vital and critical, which, of course, would lead to more fertility. The specific mutations in RB1. Some of the common specific mutations are 1BP insertion of T in exon 20. This causes a frame shift resulting in disrupting protein function, which is not good. And then G to A point mutation in exon 14 brings a premature stop to a codon, so it results in unregulated cell growth, which then results in the cancer, as you know. Then mRNA transcript. So mRNA transcript, there's the wild type mRNA transcript of RB1, which is 4.7 kilobases long. This encodes the full length of the retinoblastomia protein, also known as PRB, which, as we know, is vital for regulating cell cycle. It's tumor of sorts, regulating tumors and cell cycles. So that's why it's important to have it around, The mutant mRNA transcript of RB1. So 1BP thymine insurrection in exon 20 causes a frame shift which introduces a premature stop codon. So this basically stops the RB1, which is a problem. Then the G to the A point mutation in exon 14 introduces a nonsense mutation, which this leads to early translocation. And then both mutations produce shorter, non-functional transcripts due to early termination of the translations. So then the RB1 doesn't do its job. It's not regulating cell growth and tumor growth, which is not good at all, and what leads to cancer. And then exons and introns in the RB1 gene. So the amount of exons in the RB1 gene is 27, and the amount of introns is 26, and is separate from the exons. The difference between the wild type and the mutant type in RB1 is the wild type RB1, there's correct splicing and a functional protein. It does its job. It limits the cell growth and the tumor growth. Make sure it doesn't get out of hand. The mutant RB1, splicing errors, there's missing or missed pieces that necessary, and it's faulty or shortened protein, which then leads to the cancer because it's no longer regulating cell growth or tumor growth. So protein product, wild and mutant type. Wild type protein product, RB1, PRB1. So the protein product of RB1 is PRB1, and the number of amino acids is 928 for the wild type, PRB, not PBR. The mutant type protein product is an abnormal PRB, less than 928 amino acids. It's shortened because of the mutation, as we learned, the codon stops it early, so it often makes it faulty. Mutation example for PRBs is 1PB insurrection in exon 20, like we've been talking about. This results in 680 amino acids, which basically means it's non-functional, or it's too shortened to do its job. Another mutation example is G over A point mutation at the splice donor site of exon 14, which results in 600 amino acids. Again, too short, removes function, which is not good. The structure and function relationships. Wild type structures and functions. Amino acids, so the structure of that is 928, full length structure, proper folding occurs in active domain. So this is kind of normal type, if you will, of the RB1. The function acts as a tumor suppressor and controls cell cycles, which is important. The mutant type structures and functions. So the amino acids is less than 928. It'll vary by the specific mutation, like we covered in the last slide. This results in a tourniqueted structure. It's unfolded and it can be missing domains as well, which is not good because of that single codon that gets stopped. The mutation leads to a non-functional RB1, which means it results in a can't regulate cell division. So the loss of function because of the mutation. So yeah, it promotes unregulated cell growth, which then leads to the cancer. Disease mechanisms. How an RB1 mutation then leads to retinoblastomia. Mutations cause a loss of the RB1 protein function, which then gets rid of a viral break on the protein cycle. This leads to a nonstop cell production, especially in the retina of the eye. So that's where we see the cancer, because cancer is really just too many cell productions. It's too many bad cell productions, which this, of course, results into retinoblastomia.
So to understand how this gene works, it we need to look at the molecular mechanism. This diagram shows the main steps. The RB1 gene makes the PRB protein, which helps control the cell cycle by stopping a group of proteins called E2F from turning on the genes needed for cell growth. Normally, PRB holds onto E2F and keeps it in check. But when a cell gets a signal to grow, other proteins add groups of phosphate to the PRB, causing it to let go of E2F, which then starts the growth process. If the system breaks down, the cells can grow uncontrollably, leading to cancer.
And then these are the references we used for this presentation. Thank you.